I'm very excited to get started. Our first session is biliary tract cancer care across Canada. And I'm thrilled to introduce our first speaker, Dr. Ravi Ramjeesingh, who is a medical oncologist in Nova Scotia Cancer Centre. And he's going to talk about the Canadian biliary tract cancer landscape. Dr. Ramjeesingh specializes in the treatment of breast cancer, hepatocellular cancer, pancreatic and cholangiocarcinoma. And his research focuses on translational and health services research, as well as oncology education for residents and medical students. So welcome and started. Thank you.
Great. Thanks, everyone, and thanks to C3 for inviting me to come and talk. I didn't realize when I agreed I would be first. Yeah. Okay. So I'm going to be speaking about the Canadian biliary tract cancer landscape in Canada. And really my objective is to kind of talk about biliary tract cancers, talk about some statistics that are out there, review the landscape of systemic treatment briefly, and then actually review some of the real-world evidence in Canada that is out there. These are my disclosures. So because I'm a medical oncologist, it's ingrained in my DNA to present a case. So let's just do a case. So JPM is a 53-year-old gentleman, had a 1-week history of painless jaundice, preceding a 1-month history of abdominal pain, presented to emerge in July 2023. No real past medical history, family history positive breast cancer. So underwent a CT of the active pelvis. And you can't really see it that great, but there was a 2.3 by 1.9 by 3.2 centimeter lesion in the distal common bile duct, as well as 6 small low-density hepatic lesions, the largest 9 millimeters. So this wasn't looking too good. Their exam was normal. Their CA19-9 was quite up at 4,250. And they had an ERCP with brushings, very suspicious for malignancy. CT chest was negative. So in October 2022, there was a new patient support program and a new treatment option, which is the addition of durvalumab to cisplatin. And so we put them on that. He had 8 cycles of the combination and then did 4 more cycles of maintenance durvalumab. And we were quite pleased to see that his CA19-9 did this. So it went from as high as 4,500 and went down to baseline in the normal range. So we subsequently did a repeat CT in March of 2024, and the liver lesions had vanished. The lesion that was there, we really couldn't see a lot based on imaging scans. So I went to the surgeons and I said, hey, come on. And they were saying, all right, fine, because they just don't want to hear me complain. So they actually went and did a Whipple surgery. So they removed part of the pancreas, the common bile duct, and a bunch of other pieces of the person. And when they looked under pathology, they actually did not see any cancer. It had a complete response to neoadjuvant treatment. So T0, N0, which is phenomenal to see. And so I just saw him 3 weeks ago with his last CT scan, and it's still negative. So it's been almost a year. So it shows you new treatments can work, and there is hope. And you keep re-bringing these patients back to the surgeons when you have the opportunity. And if you're a pain like I am, they'll usually very quickly say yes. So, okay. Now, with that being said, let's talk about the biliary tract system because it's good to understand. It's an accessory organ that's part of the digestive system. It consists of the bile ducts and the gallbladder. And really, the primary purpose of it is to drain waste from the liver into the duodenum. Remember, the liver is the big filter of your body. It helps in digestion, controls the release of bile, and the gallbladder serves as a reservoir for your bile. So when you think of bile, it's produced in the liver, it's in the left and right portions of the liver, and it travels through the right and left hepatic ducts. These are going to merge into the common hepatic duct. And then the cystic duct, which comes from the gallbladder, will then merge into the common bile duct, and then it pops in into your GI tract around the inferior vater. So that's kind of the anatomy of what we're talking about. Because they're all linked, we historically has always thought of it as one cancer, but we've come to realize now that really it's a basket term. It's a cancer that has distinct tumors by location, by genomic expression, and by etiology. And so we classify it in intrahepatic within the liver, the gallbladder, and the extrahepatic outside the liver. And the extrahepatic is subdivided into your perihilar and distal. So that's kind of the nomenclature when we talk about biliary tract cancers. About 20% are the intrahepatics, about 50% are the perihilar, and another 20-30% are extrahepatic biliary tract cancers. When we look at the epidemiology, it's about 3% of all GI tumors. It's about 10 and 20% of hepatic cancers. It's the high incidence in Thailand. It is increasing in the Western countries. And unfortunately, when you look at mortality, it is also increasing over time. It doesn't matter where you're looking at, which country it is increasing. So let's talk a little bit of statistics. People who know me know I love sitting in the Cancer Society's website and point out the statistics and chopping it up. So this is a little depressing, but 1 in 2 people are going to develop cancer in their lifetime on average in Canada. So yeah, it's pretty prevalent. And then when you look at the geographical distribution of cancer, on the left is the prevalence or the rate of incidence. And you can see the darker the blue, the higher the prevalence is. And then on the right is the mortality map, which is, again, the darker the blue, the worse the mortality. So coming from Atlantic Canada, seeing a lot of blue on the left and a lot of blue on the right is not a good thing, because that suggests that there's a high incidence and a high mortality. In Nova Scotia, where I'm working in, we're number 1 for incidence and number 2 for mortality. And it is going up. When we talk about liver cancer, which the intrahepatic carcinomas are part of that population, we can see the rate of cholangiocarcinomas or of liver cancer is about 4,750. We don't have great data about biliary tract cancer. When you estimate the incidence in Canada, it's about 2.35 cases per 100,000, which works out to 936 cases in Canada. We know liver cancer rates are going up across Canada. This is over time. When you look at the cancer statistics, you can see it's going up. Part of that is because of the intrahepatic cholangiocarcinomas. You're noticing I'm not really talking about biliary tract cancer, about rates, and there's a reason for that, because no one collects it. We don't have that data. And so intrahepatic cholangiocarcinomas are lumped into the liver category. Everything else is lumped into other. So we actually do not know what the rate of biliary tract cancer is in Canada. Excuse me. And so it's unfortunate, because it would be good to know what the rates are, so we have an idea, which is why we get into real world evidence, which I'll talk about a little bit. We know there's been a lot of hope because we've had an evolution in how we're treating these patients. Historically, it was chemotherapy, even up until like 3-4 years ago, combination of gemcitabine, cisplatin. And then in 2023, we were able to show that immunotherapy works with addition to durvalumab, and then 2024 pembrolizumab. And so these are more now the standard of care that we're looking at. But again, as I mentioned before, biliary tract cancers are a makeup or a basket of multiple cancers, because we know intrahepatics, extrahepatics, and gallbladders all express genes differently. And so you look at intrahepatics, they have higher rates of FGFR, IDH1s. Extrahepatics have higher rates of KRAS. We look at the gallbladders, they have higher rates of HER2. So treating them the same doesn't necessarily make sense if the mutations and the genes in them are different as well. We also know that if you can do precision medicine on patients, they actually do better. So there's some data from intrahepatic carcinomas from 2022, where those who had mutations and were able to get on a targeted treatment for those mutations, they did better if they're on standard of care treatment. So knowing their mutation burden is actually quite important. So part of this has been built into the Canadian recommendations. This is an awesome article. No, certainly. But this was published in 2023, a guide for systemic treatment for biliary tract cancer. And precision medicine, looking at genomics, is part of what we suggest. In fact, first-line treatment, combination chemo with immunotherapy, but it's right there. Do the NGS testing as well, so you know what you have access to in the second line for treatment options for patients. And this is built into the ASCO guidelines, into the ESMO guidelines. We're not the first. So how do we get the evidence to support all this? Well, it comes from clinical trials. This is where we figure out what we're supposed to do for patients. And just so everyone's on the same page, a clinical trial is a research study involving human participants that evaluate the safety and/or effects of one or more interventions on health outcomes. These interventions could be drugs, could be vaccines, could be radiopharmaceuticals, could be radiological procedures, it could be pretty much any intervention, really, to try to determine, is there a benefit? And then the data from clinical trials are going to be used to support for approval of the drugs or the intervention compared to other things as well. So we run clinical trials because it helps determine whether new treatments are safe and effective, determine whether new treatment has less harmful side effects than what we're doing right now, and provide access to treatments patients may not have access to. There's different phases for clinical trials. You may hear first phase, phase 1, phase 2, phase 3, and that's depending on looking at the data, looking at the number of patients, looking at toxicity as well. And then there's this phase 4, which is more or less seeing what's happening out in the world when this drug has now been approved. Why is phase 4 important? Well, because we're trying to talk about, are these trial results applicable to the patient in front of you? So to get on a study, usually it's almost like the Olympic athlete, a patient getting onto a study, very minimal comorbidities, just kind of a really good candidate for a trial. But reality is when you get into the community, that's not necessarily what you're seeing. Oh, thank you. Excellent. How does she know? So, for example, BRCA1 and 2. So this is important in pancreatic, breast, and ovarian cancer. It's estimated that the BRCA1 and 2 genes mutations are 1 in 300, 1 in 500, and there's a lot of studies looking at these rates as the thing that affected Angelina Jolie. But we get this data from things like the Cancer Genomic Atlas Program, or the TCGA, which is a phenomenal project and study that looked at a genomic program looking at different tumors, matching normal supplies for 33 cancers. The problem is there's little information on minorities or different racial or ethnic groups. Okay, so 77% of the patients were white. So we're a very heterogeneous country. So when you have a patient in front of you, how applicable is this data to the person you're seeing at when you have such a focused data set? And so this is where things like real-world evidence comes into importance. It's looking at a population and looking at what's going on in that population, and it gives you more diverse data set than running a clinical trial. And it gives you a full patient journey of social determinants of health. And really what it is going back into health records, pulling out the data, looking at hospital pharmacy data, patient registries, and trying to identify what works for patients. So when we look at Canada and we talk about what's real-world evidence, there is data from Ontario, BC, Saskatchewan, and Nova Scotia. Saskatchewan's in red because it's behind a paywall, and I couldn't pay for it. So I'm going to talk about Ontario, BC, and Nova Scotia. I'm not cheap. I just couldn't get to it. It's hard. So when we talk about Ontario, real-world evidence, so this was published in 2023, looking at just over 2,000 biliary tract cancer patients in Ontario and looking at administrative databases as well. And some of the things that you can glean out of this is roughly equal male and female population, almost 50-50. Majority are presenting late-stage disease when they're being identified. About 30% intraherpatics, 30% extrahepatics, and the rest are kind of a mix of gallbladder and ambulatory cancers. And then when you look at treatment, 80% are getting on first-line treatment, and then a substantial drop for second line to 24% as well. So a phenomenal drop in the number of patients who can make it to second line in this real evidence population. And then when you look at BC has nicely shown that, so the green line on the top is the population growth of the province. And then the other three lines are the intrahepatic, extrahepatic, and gallbladder. And you can see the gallbladder, which is in purple, and the extrahepatic, which is in darker green, I guess. It's kind of following the population growth. So as population grows, you kind of expect it to kind of increase as well. But the intrahepatics, which are kind of this color here, it's actually not following the population curve. It's actually going up faster. And that's saying the rate is increasing faster in BC than the other cancers as well. And they also showed, again, when you have first-line treatment, about 55% of patients were making out the first-line treatment. And then a substantial drop to second line, 14%. We're making the second line treatment. Now, why? We don't know. That isn't really part of the presentation of the data they looked at. But we're seeing correlation between Ontario and BC. They have very similar data. So Nova Scotia, this is not published, it's fresh off the Excel spreadsheet. Okay. So between 2010 and 2019, there was 643 biliary tract cancer. Again, similarly, 30%, 30%, 30%. It seems to be with expression, and we're seeing about 64 per year. And so we're actually in the process of going up to 2022, which will be about 73 patients by the time we're done. But what we're finding is, 40% are having pathological confirmation of the disease. 25% are having a CA19-9 that's been drawn on them. And only 47% meta-oncologists, of which 40% were ineligible for treatment. So very few are getting on treatment as well in 2010 and 2019. So, when we talk about clinical trials, we are not supposed to compare clinical trials, but we can compare real-world evidence because they're data sets. So if you look at BC, which had 15 years of data, 2015 to 2019, Ontario 10 years, 2010 to 2019, and Nova Scotia 2010 to 2019, you see the number of patients in each data set, 1,520, 2,143, and 643, which when you figure it out per year is 101 for BC, 214 for Ontario, and 64 for Nova Scotia. But then when you standardize it based on the population, so per population, so I just randomly picked 2015. You then see, when you look at the BTC cases per 100,000, it's 2.2 for BC, 1.6 for Ontario, and 6.8 for Nova Scotia. So Nova Scotia has 3 times the BTC rate than the other locations. Why? We have no idea. That's a work in progress. This is data that came out 3 days ago from my spreadsheet. So we got to look into this a lot more. But then also, when you look at who are getting on systemic treatment, BC is about 55, Ontario is 80, Nova Scotia is only about 19%. So this is why it's important to do real-world evidence, because this is how you can start to pick up patterns and start asking questions about what's going on in your institution, your province, and how can you fix things. So we had an inkling about some of this. And so with the idea of precision medicine, we know we were behind the 8 ball a little bit. And so we were able to partner up with the QE2 Foundation and the BMO Right for Cancer, and they were able to fundraise for the new Illumina NextSec, which we now have clinically for patients and since 2023. And we have a new focus panel, and then we have liquid biopsy coming at the end of the year. So we're trying to catch up to other places. But we said, let's look to see what happened with the NGS. And so it's been running since January, February of 2023, and we've had about 15 patients undergo the testing, of which we found 10 had mutations. So that's about 60%. Now, it's probably a little higher than it should be, but still, that's a lot higher than what's usually published in most places, which begs the question, what's going on here? Why is our mutation burden so much higher in Nova Scotia than other places? Well, you can see we have FGFR2, 3, IDH1, KRAS, HER2. So we're seeing pretty much the gambit of everything. So let's see what happens with the, there's some.
Do you have germline on that?
It can be done, though. Yeah. It has been done on a couple of patients, but not all 15. So then why were patients not getting tested? Because as I said, it was only 15 to 69. That's quite a lot. So, 20% didn't have enough tissue because they had scrapings, and that wasn't enough to do testing. Some 18% were adjuvant and neoadjuvant, which are usually not the ones who are doing testing in the first place. We had 18% say no, they didn't want treatment. And so they didn't want treatment, that we just didn't push the issue. 28% had poor performance status. They just weren't fit enough for treatment. So a third of our patients we're seeing aren't fit by the time we see them. And then we had a patient pass away, unable to treat, no tissue. The 22% where they wasn't sent, that's the one we're going to look at because there's no reason why they weren't sent. So we're going to look into those cases to try to figure out a little bit more about why testing wasn't done with those patients. But for most of them, there's a reason why we didn't do testing in them. And so it doesn't give us a great idea of the population itself, but it does help in the way of focusing the testing a little bit and helps on the cost as well for the path lab. So with all this going on in Nova Scotia, we try to strive to do more education there as well. And so we run a GI clinical trials meeting every year, which is more like a Dragon's Den where companies come to us. It works well. We run an HPV update where several people here have already spoken to. And then I organize the first Nova Scotia Oncology Research Day. Advice, never be the person to organize the first anything. It's a lot of work, but it allows us to get the information out there for patients. So quick plug, and we're almost done. There are a lot of precision medicine treatments for GI cancers. There's more on the horizon. But when you start looking at clinical trials in Canada right now, and so I went through clinicaltrials.gov because I have all the time in the world, and looked at all the BTC trials that are ongoing. There's only 4 in Canada for BTC. Now, there's another bunch of basket studies that BTC can be rolled into, but specifically biliary tract cancer, there's not a lot. So my plug is we need more trials. And just lastly, just for your information, the Eastern Canadian GI consensus statement, which was just very recently published, does have something on biliary tract cancer as well, if you're ever looking for information. So to summarize, biliary tract cancers, they're not as rare as we thought they were. We used to think it was rare. It's still rare-ish, but we know the amount that's out there is more than we thought it was. Systemic treatment and precision medicine, we are definitely improving the outcomes for patients, but we need more trials. And with that, thank you.
Well, thank you very much. That was very, really hot off the press data, which I love. That's fantastic. We're going to have about 5 minutes for questions before we go to the next speaker. Questions, you can use the mic. Leonard, I guess, is passing it. Actually, it would be better if you come up there because then the people online can see and hear you at the same time.
Okay.
Carol.
Doctor, I found your presentation immensely informative. I'm an advocate for my wife, Thelma. We're Eastern Canadian as well, from Newfoundland.
Mic a little closer so that.
Sorry?
So everyone online.
Oh, no, one, sorry. So very discouraged or very interested to see Newfoundland as one of the darkest blue places in the province. And I have a question. Because I'm trying to start doing some advocacy on behalf of everybody from the gift that Dr. Al gave Thelma and my wife as a result of this foundation. I'm retired. I have the time to do it. I've already started doing it. There's an election in Newfoundland this year. What a better time to do it. Wondering, with the oncology folks in St. John's, do you have any connection to any of this work that you're doing? I knew that you were heading up the trial for the IDH1. But is there anybody you're connected with in Newfoundland directly that can, are you meeting, are you getting together? When you mentioned Eastern Canadian Biliary, I'd be just curious to know, did that include Newfoundland?
It does. So, the data set is Nova Scotia, but when it comes to trials, any information, I'm talking to them always, so that they're aware.
Okay.
Newfoundland has some challenges when it comes to how much trials they can do, how much genomic testing they can do, and working on getting that improved. But as far behind as Nova Scotia was, Newfoundland was a little bit further behind. But they're making amazing strides moving forward because they have a lot of really good people there that are pushing. They've seen the benefit of genomic testing and things like that. But fortunately, everything is like time and money and getting the resources. So advocacy is key. Because I hate to say it, people don't listen to doctors as much. They do listen to patients. And so that advocacy is extremely important because I could go up to government and say, we need this, and they're going to go, yeah, sure. And it won't happen. But a patient going up and saying that, they will listen.
I'd like to talk to you some more. I won't keep the podium in case somebody else has a question within the 5 minutes to discuss it more. But you're right. The doctors are probably too busy. They're constrained by money. And I'm just letting you know that I'm just across the pond. I want to start helping you help us.
Very right away.
Thank you. Enjoy your presentation.
Do you have any other questions? I think the point about advocacy is so key, but I actually think if we do it together, patients and clinicians and researchers, we actually have the strongest voice possible. But I completely agree with Ravi. Like, you go and you talk to a politician, they said, data's okay, but I want to talk to a patient. I want to know why this matters. And we don't remember data, but we remember stories. And so I think that the value that you as patients in the audience here bring to that conversation absolutely cannot be underestimated. And when we tell the story together, it's so much more powerful. And thank you for everything you're doing in Eastern Canada. So. That, I will move to the next speaker. So it's my pleasure now to invite Dr. Rob Grant up. He's a medical oncologist at Princess Margaret. Dr. Grant specializes in biliary and pancreatic cancer treatment. His research applies artificial intelligence to clinical and genomic data to improve cancer outcomes. He leads AIM to reduce and co-leads the LeGresley Biliary Registry, and the LeGresley family has generously contributed to the meeting today, and PancureX Translational Research Initiative. So welcome.
Thank you. Thank you, Rebecca. Thank you, Leonard. Thank you, C3, for the invitation. I was invited to talk about the LeGresley Biliary program as a model for something that really resonates when I hear Rebecca talk about it, which is research is care. Several conflicts of interest. And what I'll discuss today is an overview of the program. And then I'll focus on one of the technologies that really, I think, exemplifies this idea that research is care, whole genome and transcriptome sequencing. And then I'll provide a kind of whirlwind of examples on how we're impacting care, improving outcomes, but also advancing the science. And as Rebecca said, the LeGresleys have supported this meeting, but they're wonderful people. And they've been huge supporters of cholangiocarcinoma and biliary cancer. Their family was affected by biliary cancer. Their son, Richard, in 2018, they lost their son, and he would have been 33 just last month. So out of that, they've been really transformative supporters at Princess Margaret in terms of advancing the vision of research and care for people affected by biliary cancer. And we share their vision that something like that, hopefully there's a future soon where something like that will never happen. And we talked about advocacy and research and care. Another thing that's really critical in the Canadian landscape in particular is this idea of philanthropy. And their gift was transformative. Their sustained support is really what makes everything I'll talk about today possible. They've also attracted, it's a cycle, and they've attracted other donors into the fund. We do the ride every year. I'm going to join this year. I'm a little bit terrified, but it's one of the major sources that helps fund the research that we do, and also there can be gifts given through Princess Margaret Foundation. And the LeGresley family shares this vision that this shouldn't happen in the near future. And so, what's the vision of the program itself? And it's really meant to be a link for patients to the best clinical care in the world. So everything that the latest technology and clinical trials has to offer, so people get the best outcomes, but also have the opportunity to contribute to scientific advances that lead to better clinical care and this virtuous cycle. Richard, their son, was affected by a condition called primary sclerosing cholangitis. This is an autoimmune condition that causes inflammation in the biliary tracts. It's one of the conditions with the very highest risk of cholangiocarcinoma, biliary tract cancer. So in part because of our expertise as a center, we have some world leaders, Gideon Hirschfield and others, but also because of that story, we have a focus on PSC. And this is a small picture. It probably could fill the whole screen now, but it's only possible. I'm here just representing a big team of really dedicated people to this vision. There's a whole bunch in the audience here. You'll hear from Arndt and Jennifer later today. Programs co-led with Gonzalo. Can you see my cursor here? No. He's in the white shirt there. But clinical fellows, managers, coordinators, bioinformaticians, fellows. It really takes a whole village to do this sort of work. So what is the program? So the typical process is one of the clinicians will introduce the program in the registry, and then a coordinator will come in and talk about this process. And what patients are contributing is clinical information, so high quality data about exposures in the previous life, things like smoking, but information so we can understand how the cancer may have developed. But then rich real world data. I had Ravi's slide, which was very helpful for this talk. So he's gone over real world data, contributing real world data longitudinally, but then also biospecimens. So tissues and fluids that we can use to study scientifically. So if there's a surgery, some of that surgical specimens, we do specific biopsies for this program. And then we collect blood and stool. All of this is banked, but it's not meant to sit in a bank. It's meant to be analyzed for clinical care, but also to advance the science as quickly as possible. And we do all this serially because we want to understand how do these cancers change over time with treatment. And this program predated me, but as I joined, there was this really wonderful merging of different forces, one of which was the Terry Fox Marathon of Hope, which is a national effort to really bring Canadians into this genomic world, where whole genome and transcriptome sequencing paired with high quality clinical data is meant to be collected over short years on many thousands of patients. And we've been one of the largest contributors here in Toronto to this program. The biobank, we have lots of, I'm going to talk about whole genome and transcriptome sequencing because I think it's the best example, but these specimens are already being used. We have single cell spatial data with Martin Jackson and IMC about to come soon. Valentina's in the audience somewhere here doing plasma RNA sequencing and some really cool work. Tracy McGawha's looking at the stool. We've got metabolomics. So I'm really just highlighting one element, but the idea is that this is really a catalyst for really broad, innovative research. So whole genome and transcriptome sequencing. So we've already heard about the excitement around precision medicine in biliary cancer, and I know this is an audience that come from different backgrounds, and so I thought I'd just highlight what this really means. So there's this concept, the central dogma of biology, and it's pretty amazing. It's a simplification, but it's a really powerful idea. We're all made up of cells, 30 trillion cells, and each of these cells has the same blueprint, the same code of our life, of us. And the code is fairly simple in its most basic forms. It's just this repeating pattern of 4 molecules, A, C, T, and G. And from that code, that creates us. It gets transcribed into this messenger molecule, RNA, which can also be studied. And then that's translated into proteins. And these are the building blocks of life, structure and function. It's what makes us, us. But mutations in the DNA, changes in this letter code, also much more complex changes than just substitution, but mutations in the DNA change the proteins, the structure and function. And that's what causes cancer, again, in a kind of reductionist view, but this is something we've known for a long time. And precision medicine is looking at an individual tumor, those individual mutations, and targeting it with a specific drug. So one tumor is treated differently than another based on what the genetic or other molecular makeup of the cancer is. And so looking at the program today, there'll be lots of discussion on DNA sequencing. And I think C3 and others, but particularly C3, has done an amazing job for advocating for targeted NGS, next generation sequencing panels. This is not standard or routinely funded in Canada. It should be. It's advocacy. But what that's doing is sequencing the key genes known to drive cancers or have existing drugs to target those genes. And it's about 1 to 2 million base pairs of those ACTG. But thankfully, every year there's new targets, there's new genes. And so these panels keep expanding. And one of the things I look at when I meet a patient is if they've had sequencing, what's the state of the art? Is it what we currently look at? And so another technology can look at the exome. This is all the genes, all 20 to 60,000 genes, depending how you count it. That's 30 million of these base pairs. And that's another great technology. But to get a full picture of what's happening in the cell, we want to look at the whole genetic code. And the genes are only about 1% of the genome. And we know that this other code, much of it is important and affects the function of the cells. So we focus on whole genome sequencing. And it's a complex process, right? There's the technology itself, but the technology is only one step in this process. And this is a slide from Jennifer Knox and Anna here, so thank you. But this was a process when we started the biliary registry. We had about a decade of experience, or 5 years at the time, of doing this for pancreatic cancer, which is where it started. And about 10 years ago, with the COMPASS trial and the McCain Center and PanCure here at OICR, we optimized this really complex process that involves dozens of people going from that coordinator that I talked about and the patient to the data collection, to the biopsy, through all the different hands, the biobanks, the laboratory medicine, through to the sequencing technology, the bioinformaticians, to get this clinical report. And at the time, it was thought to be impossible to do on a liver biopsy. And so we were able to do it within 8 weeks, which was amazing, but we've been iterating since then, and I'll show you some of that. And Ravi's showing how quickly this can change, the situation can change in biliary cancer. So we want to be faster and faster. We've been working on something we're calling ultra rapid whole genome and transcriptome. We had a time limit, but it keeps going down further and further. And again, this is primarily driven in the PanCure pancreas program, but we're hoping to bring it across to biliary very quickly. Again, a collaboration between OICR, McCain Center, and the Biobank and others. And we've been able to do this in a time frame with much more complexity. I'll show you some of the changes to commercial assays, but in a range that's comparable to what some of the really massive American companies offer in terms of turnaround time. So in a really ultra rapid format, and we're hoping to continue to improve on this. So in addition to the whole genome and transcriptome sequencing, which gives us this really complete picture of what's happening in the cancer cell, one of the challenges is getting a good specimen. I suspect when Ravi goes into some of the data, what they'll find is that there wasn't an area that could be safely biopsied, or when they did biopsy, that there wasn't very many cancer cells. He saw that in the surgical specimen. And so we've been big believers in this technology called laser capture microdissection, and we've really applied it almost universally, except trying some cases without it. And the idea is, this is a pancreas cancer, but the biliary cancers are the same idea. It's a mix. When you get a biopsy, it's not just cancer cells, it's immune cells, it's connective tissue. And if you're focused on finding the cancer mutations, everything else is noise. And so what we do is have a pathologist annotate where the cancer cells are, and then burn it out with a laser so that we can sequence that area preferentially. And it makes a big difference. So here, this is called a violin plot. On the left are cases that have had this laser capture procedure. On the right are those without. And what we found is that the cellularity, the proportion of the tissue that we've analyzed that's cancer versus non-cancer, is much, much higher. It's enriched through this process. And without LCM, at least in our program, when we try to do this process, there's half of the cases fail because it's below a 30% tumor content threshold. And so we can't clinically report what variants are there. So it's a critical process. And importantly for the patient, it translates to finding more targets, to being able to potentially inform their future treatment plan. So that's the technology. So we believe in this really high quality, comprehensive view of the cancer cell, but also in scale. So Ravi introduced the TCGA, that's the American effort, 14th province. England has a similar initiative. I mentioned Canada is starting to grow. And because this is a rare cancer, biliary cancer has only been a small fraction of these large sequencing efforts, less than 100 patients. And this idea of laser capture whole genome sequencing has never been done at any sort of scale before. And what that means is there's probably genes that are driving 10% or more cancers that we just don't know about, right? And so the hope is as we grow our program, you'll see the numbers shortly, that we're able to do that and find these new targets, develop new drugs, improve care. And so this kind of convergence of the marathon of hope and the kind of the scale, initially we were focusing on young patients with PSC, because that's what we had the resources for. But all of a sudden, everything changed, and we just decided we're going to do everybody. We have the resources and the ability to do it, and the program's growing really rapidly ever since. And right now, we have over 300 consented individuals to the program. The majority are contributing whatever they can: blood, stool. I think a measure of how dedicated patients are to a program is how much stool we get. Went into more detail. But we have sequencing on over 150. I think that this is the largest, it's not one of the largest whole genome and transcriptome sequencing data sets. We're going to be sharing it very broadly through the Marathon of Hope and other mechanisms. And the only that I'm aware of that does this important laser captured step. Arndt, you'll hear from later. He's developing what I think will be a really impactful program, growing these cancers either in mice or as organoids so that we can test drugs, understand more about biology. So I think that'll be a really exciting aspect. But I'm going to just highlight some more about how whole genome and transcriptome can impact clinical care and some of the scientific advances. And the most obvious impact on clinical care is the molecular rounds that we have. And this is similar to what C3 is doing, but every case we have every other Wednesday, we review the results as they come in and make recommendations on how it could impact care. I'll just share one story that was an update at our last tumor boards, but I think is a good example. It's quite technical, but what we have bioinformaticians, clinicians, a whole team reviewing the data as it comes in. We tell the story that this individual has a history of Crohn's disease, which is an autoimmune disease that's linked to PSC, although there wasn't known PSC, and had an intrahepatic cholangiocarcinoma and had started chemotherapy without immune therapy because we were worried about the impact of immune therapy in Crohn's disease as we started. And when we got this result, you can see quality metrics, germline, which I'll talk about in a minute, and the different individual mutations. But the most striking thing wasn't an individual mutation like you might see on an NGS panel, but the broad mutational signatures and a metric you would get from an NGS panel, which is just overall there was an enormous number of mutations in this cancer, a really unprecedented number of mutations in this cancer. When we look across the cohort, the x-axis here is how many mutations are in a biliary cancer, and it's typically this rate of 1 to 10 mutations per megabase. In this cancer, this is a log scale, so is an order of magnitude above all other cancers, including some of the ones that we have known causes for hypermutation, like Lynch syndrome you may hear about. And this had an impact on care. We weren't giving immune therapy. Hypermutated cells, the proteins on the surface change dramatically, so they're more likely to be recognized by the immune system. And so we can use immune therapy to kind of kick the immune system into gear to try to have the cancer cells attacked by the immune system. And so with these results, we started the immune therapy. This is the CA19-9 number, which was rising, came down along with shrinkage on the scans, and there was a sustained response. This serial profiling, which I talked about before, so eventually there were some areas that were growing, some of which looked different than others. And we sampled one of the different looking areas, and this same really striking hypermutation was present. But the most unexpected and still unexplained feature was that all of the individual mutations were different. And so what we have here is two separate hypermutated intrahepatic cholangiocarcinomas in the same person. I've asked many world experts on this, and we've never seen this before. There are scientists here trying to figure out what's driving this. Was there some inherited or other factor? But again, it informed care. The cancer was growing a little bit, and we switched the immune therapy to a dual immune therapy, and we're able to regain some control and shrinkage. So that's one story, but each of those now 150 genomes, transcriptomes that we've done has their own story. And we just published our first 50 or so cases in the Canadian Medical Journal, Oncology Journal. And what we found was that a quarter of people have really have targets that are OncoKB1. They clearly can impact future treatment plans. Half or more have some sort of change that likely, we believe through our tumor board should be considered in future treatment plans. But to Ravi's point, when we look at people who actually traveled through the journey and needed second-line treatment, so the first chemotherapy hasn't worked, they needed another treatment decision. Among those with targets, half were able to receive targeted treatments. But at the time, some of this has changed thanks to people in the crowd here, but none of them were publicly funded. People were either paying or they were accessing it through compassionate programs or other similar mechanisms. So I think this is changing in large part to advocacy, but obviously is a problem for equity and other aspects. And those targeted treatments for some people worked really well. So we have just on the right here a CT scan showing for an FGFR2 inhibitor, somebody's cancer had grown on chemotherapy, started this pill, and had a sustained shrinkage of the cancer with this pill with less toxicity. So that's clinical care, but there's really this virtuous cycle with research and back and forth. And one of the projects that we're most excited about, proud of, because of the origins of the LeGresley program is studying specifically primary sclerosing cholangitis and the cancers that are associated with it. What makes them different, and can we come up with better treatment strategies? And so because this is a rare subgroup in a rare cancer, we combined with some collaborators at MD Anderson to generate a larger clinical cohort, but then we analyzed our genomic data. This was led by one of the oncologists now, Kevin Wang, as well as Felix, who's here in the crowd. We'll keep seeing his picture. He's our lead bioinformatician. And I think we learned some important lessons. The genomic landscape of PSC cancers is different. They don't have the same targets, although there are some different targets. But this tumor mutational burden, the Crohn's disease association may have had some impact on that hypermutation. 6 of the 16 PSC-associated cancers had a high tumor mutation burden too. Not in the same range over 10, but more than we would expect typically. And then the genome-wide signatures, we can look across those 3 billion base pairs, and we can see the kind of signatures, the mutational processes that seem to give rise to these cancers are also quite distinct. And maybe not surprisingly, because this is thought to be, at least in part, an immune condition, the immunology of the cancers are different. And we see this kind of unexpected, but probably explainable phenomenon. On the top here, the orange cancers are the ones associated with PSC, the blue not. And we see this kind of bimodal distribution, where PSC cancers are the most immune activated. This is also seen on transcriptional programs. These are immune cells, but also the least immune activating. So maybe they're kind of burnt out or activated. And we think this may have some implications on treatment. But we were able, through the combined clinical data, to ask, does immune therapy help these individuals? And because of the autoimmune link, I think many clinicians have some nervousness about using immune therapy in an autoimmune condition, that it might activate and cause a flare and cause injury. What we found was that it was safe to do. There were a few small flares, but maybe not more than what we would expect just as part of the natural history. But there was small numbers, but I think a suggestive signal that immune therapy helped delay progression of disease and was more likely to lead to a response. So that's one story. We have several others. I'll just highlight 2 in the interest of time. But another is inherited causes of biliary cancer. So Steve Gallinger, a big mentor of mine, showed that BRCA, which Ravi highlighted in his talk, also causes pancreas cancer. He learned that in the 90s using real-world evidence. And then we looked more broadly, and we were able to show in Ontario that more than 5% of pancreas cancers have an inherited cause. And you can't figure out who it is. And that 5% threshold is important because it's what the government uses to say, we'll pay for an inherited genetic panel, a test. And so for the past few years, in large part because of that research, every person with pancreas cancer that we meet has the ability to be tested. Is this an inherited cause? If you find a mutation, it has a huge impact. Individuals in their family will carry the same mutation, pathogenic variant is the proper current term, and can undergo cancer prevention, preventative measures or screening. So it makes a big difference by preventing loved ones from being affected by cancer. Because we had this genomic data, we also get the inherited. We sequence the blood to know what's in the cancer. And we started to find that there was more than 5% of biliary cancers, which there's a few small studies suggesting this may be the case, but I think is really not widely appreciated in the field. So we just started sequencing everybody, looking at the inherited causes as well. And what we found was the same thing. More than 5% of individuals have an inherited cause. Actually, 14% have an important mutation, a pathogenic variant in a cancer gene, but more than 5%, their cancer seems to have been caused by an inherited pathogenic variant. And we know that because we can look at what the mutation is and what sort of signatures we'd expect to see in the genome, the tumor data, and then combine those together to understand that the cancer itself carries those signatures and likely it was a causal mechanism. So we're meeting with Cancer Care Ontario, hoping that we can make this case, and we'll hopefully publish this soon so that this can expand, hopefully to become standard of care across the world. So those are more on the clinical end of translation. We're also working on the more basic elements, trying to understand like how do these cancers develop, like what's the underlying biology and science. And Felix has really been leading a project because we have this really unique, high quality data set. We're able to ask questions that have never been asked before, like what are at a really high resolution, how do these cancers develop and evolve over time? And this plot on the left is just showing because of this enrichment, the laser capture, we're able to, with really high sensitivity, detect copy number changes, big changes in the chromosomes, and find genes that are amplified or deleted. And because we have the expression level, we're able to look, does this actually change how much of the gene is expressed and presumably its function in the cell? So we're able to integrate all this data together to confirm known genes, but also to discover genes that may play a role in cancer that aren't previously appreciated, as well as some really cool, more complex things like extrachromal DNA. So one of the genes seems to be creating these little rings outside of the usual chromosome, and that's a mechanism that it gets amplified. And then we're able to infer the evolutionary patterns. We believe that there's at least two big branches. Some of the cancers are driven by a BAP1 deletion, others or mutation, others by a P53, and that leads to different co-mutations, different genomic signatures, different gene expression programs. It has anatomic associations. The BAP1 is more likely to be intrahepatic, the P53 more likely to be extrahepatic. So we're starting to explain the clinical and anatomic heterogeneity. But likely these cancers need different treatments because they seem to also have different natural histories. So we're really starting to understand some of the underlying science. And so with that kind of whirlwind, I'll just pause to thank C3, but also to point out C3 is really growing rapidly over the past couple of years. The LeGresley program, there are some other programs nationally that have experienced similar growth. And I'd just like to highlight that I think Canada is kind of rapidly becoming a world leader, both in clinical care, thanks to advocacy, thanks to everything that's going on here, but also in research. And I think it's time that we have an opportunity in Canada to step into this leadership role, to drive the science, to drive care, and ultimately this helps everybody affected by cholangiocarcinoma and biliary cancers. So thank you to everybody involved. Wouldn't be possible without patients and their caregivers. I've highlighted LeGresley. There are other donors as well. Funding agencies, OICR, it's fitting that we're here. This is where all the magic happens, upstairs and the floor above. And the clinical and research teams at Princess Margaret and at OICR. So thank you. Happy to take any questions.
Thank you so much. I'll, while people are coming up to the microphone, if there are any questions, and please, like Leonard said, there's no stupid questions. So please feel free to ask questions, but I'll just make a comment that, my husband is lucky to be part of the LeGresley program as well from a research perspective. And even from the moment that I reached out to Steve, Rob's mentor, started the program, and Rob and Anna and all the individuals in LeGresley, it was never a matter of should we collaborate together as C3, but it was like, how are we going to do this and how can we do this the best way possible? So I think this is very much as the spirit of what we have in Canada is like, we're small enough that we know we have to work together, and how do we do it the best way possible? So I think it's just an example of why we can be world leaders. There's this no interest in competition here. My husband has not given a stool sample, so I just want you to know that should reflect his interest in the program, but it also occurred to me that maybe that is where the expression "give a shit" comes from. There you go. Do you have concession? Yeah.
I have a quick question. You showed the LCM capture data and how the samples that didn't have LCM had a 50% fail rate.
Yes.
I wonder what the fail rate was for the ones that did have the LCM, if you know that.
15%? That's not right. Felix.
It looks pretty.
Yeah.
Less than 15?
Less than 10% maybe. Yeah, somewhere in that range.
Great, thank you.
Yeah. And it's not something any of the commercial, it takes that slide that I showed of the complexity of the program. That's one of the most complex steps. So it's something that is very difficult.
So my comment comes to what Dr. Ravi was saying. I'm in the Ottawa area. I'm a patient. How do I get involved? How do I contribute? Whatever specimens. What do I do?
That's a great question. I should have been prepared for that one. We try to see as many people as possible. We're a big team, but a relatively small team. But we send referrals to us, and we'll try to see people.
So through my doctor? Is that what you're saying?
Correct. Yeah.
Thank you.
We're also looking at ways. Rebecca has some sequencing programs through the Marathon of Hope. The idea is that this is really a national network where individual centers. I know George Zagopoulos is leading a similar program in Montreal. So this is every program is unique in its own way, but there is a growing network. So I think speaking to your doctor, wherever you are in Canada, but what sort of initiatives are available is always a good thing to do.
Thank you.
I did want to make one point because just for anyone, for patients in the audience who might be confused. So we talk about molecular testing, genetic testing, genomic testing, and we use often those words very interchangeably. But I think Rob mentioned earlier, sometimes we say somatic testing. That's testing on the tumor. So your tumor has a whole bunch more mutations than the rest of the cells in your body. And that example of there's so many mutations per cell. So that's testing in the tumor. So when C3 offers testing, we're offering testing on the tumor. Now, sometimes there's mutations in the tumor that we also know that you can have in every cell in your body, like BRCA, for example. And so what Rob was talking about in one part of his talk was around testing the blood. So if you do the same test looking for mutations on the blood, those are germline mutations. Those are things that you have been born with and you could potentially pass to your children. And that's why it's so important, because if there's a mutation in your blood that predisposed you to getting that cancer, then you probably want to know that and potentially want to be able to have your family members tested. So there are two different things, but what we're offering with C3, the tests that we give you do not tell you that your other family members are at risk, but it can sometimes give you an indication that, as Rob says, you may want to get testing, and you're really advocating for people to get testing if they have those specific things found.
Yeah. And one thing I'll say is that the testing from C3, I spent a lot of time looking at the details of one test from another. The testing that C3 has arranged is fantastic in terms of its quality. So it's there is some variability in terms of commercial offerings in particular, and this is a very good test they've selected.
Yeah, and we're very grateful to UHN and OncoHelix who are supporting that testing, and we're having a session this afternoon on that as well. Well, thank you very much,
Great.
Rob. Really
Thank you.
appreciate it.